So hello everyone. Today we're just going to go over two units, pregnancy and fertilization, and then the steps of childbirth and whatnot. And then later we'll move on afterwards for the cardio unit after. So fertilization, it's a fusion of gametes, and this creates something called a zygote. And the zygote is basically going to be what turns into the baby, the embryo in the end. So the first we have to know is that you have many sperm. You need many sperm to penetrate the oocyte's membrane. And this is called the corona radiata. So you have to have multiple sperm. This is because of you need the acrosome to break through the follicular cells then. And then after this for breaking, the fertilized oocytes becomes a zygote. And then the zygote will begin division. After a zygote is formed, it will undergo a process called cleavage, which you can imagine is kind of like mitosis, where it's going to split in the middle and then split it again. And then after cleavage, it turns into a blastomere. After many forms of division, you get the morula, which is a solid blast of cells. The morula then becomes a blastocyst and hatches from the zona pellucida, which is a hollow ball with an inner mass. You kind of imagine as this. Instead of getting this typical division, it gets like that. And then the blastocyst will go into the functional layer of the endometrium. It'll attach onto the functional layer. And the outer layer, the trophoblast, the outer layer is known as a trophoblast, which will later become the placenta. The trophoblast will help lodge the blastocyst within the endometrium. And this is a process that we know as implantation, and this leads to changes with both the trophoblast and the inner mast cells. The inner mast cells to provide nutrients to supply the maternal tissue, and you have the creation there as well. The inner cell mass also turns into, I should say, the amnion and the yolk sac. We'll go over this later. And then after they undergo a process called gastrulation, gastrulation of which this trophoblast turns into the ectoderm, the mesoderm, and the endoderm, which all leads to different sorts of tissues in development. The big idea is you need to know that they have different ideas. Well, they lead to different tissues. There's a general list you can look online, but the gist, I won't say them out now because there'll be way too much to do. So, four embryonic membranes form around the embryo. The amnion creates a cavity, which is basically like the space for development. Yolk sac, we can say, is for nutrients, nutrient collection and distribution. And you also have structures called the chorion, which separates blastocele from trophoblast, meaning basically like a layer here, you can call it the chorion. And then the allantois will form part of the umbilical cord. So what you're going to see is you're going to see like a yolk sac that looks like this. And then the embryo is going to develop here, let's say, attached to the yolk sac. And then you're going to see this is going to be known as an amnion, the space where it develops. And then this outer layer will be known, this layer. This layer will be known as a chorion. And the squiggly thing on the outside is going to be eventually go into the umbilical cord. Well, it's going to be called the allantois. And interestingly enough, there's two ways that we actually use this information for health benefits. You can use Coriolis villi sampling, CVS, or amniotic fluid. These you can test for stuff such as like what's it called? Anything such as Down syndrome, any genetic issues with the child, any developmental concerns, or whatnot. And well, there's also the question, which one should we use? Well, generally, if it's in first trimester, you would use CVS. This is because the chorion is present during first trimester, whereas amniotic fluid develops later in second or third trimester. So you just need to know that if it's earlier, you would likely use the chorion instead. Now let's talk about the placenta. The placenta, imagine if you have a pregnant mother and she has all her lungs, her digestive organs, her heart, and whatnot. She has to supply nutrients to the developing baby, the embryo. So the placenta is an organ that separates this, that acts as an interface for nutrient and gas exchange, eliminates waste products, and secretes hormones. Well, let's talk about this. Trophoblast, the structure of the placenta is a complex structure with parts of the chorion, as we mentioned, and also the trophoblast. Trophoblast cells surround, let's see if we draw it out. Trophoblast layers surround the villus of the placenta, which are responsible for the hormones. So imagine the placenta is very hairy with a lot of villi here. And these villi are derived from trophoblasts. They secrete hormones. And the chorion forms the villi. Well, I guess I should say the trophoblast layers surround the villi, but the chorion is the villi itself. Remember how we talked before? The chorion separates basically the chorion was a layer that separates the embryo and the space from the umbilical cord. You can just think of it as space, but you can just think of it as a layer there. During early pregnancy, the embryo, or specifically, the embryo secretes HCG, and HCG, remember from before, acts on ovarian tissue, signals a corpus luteum in this aspect. Corpus luteum to suggest progesterone and estrogen. HCG is similar to LH, you can say, similar to this. And this is one of the earliest detections of pregnancy. So the trophoblast, I should say the trophoblast secretes HCG, and this acts upon corpus luteum. And yeah, this progesterone helps thickens the uterus and maintains endometrium. In doing so, this ensures that the trophoblast is properly implanted in the endometrium. And if it's not properly, if we don't have HCG, no progesterone, then the trophoblast will not be implanted and it'll just be shed. So before the end of the first trimester, placental cells take over the secretion of progesterone and estrogen. What you can imagine is you split it into three trimesters. We can say first trimester, second trimester, and third. Initially, you'll have a great peak of HCG, That'll slowly just plateau. And then after, well, you'll see it initially in first and second as well, but you'll see increases in estrogen and progesterone, but progesterone will drop. So let's label this HCG, estrogen, and progesterone. So during the first 2 months, the corpus luteum degenerates, but secretes, uh, the trophoblast secretes uh, progesterone. Remember? So let's see. Trophoblast secretes HCG, which acts on corpus luteum, progesterone, and this maintains the corpus luteum, maintains the endometrium, and then the trophoblast also secretes estrogens. And yeah, and these will be used later in development. Let's see if there's anything else. Yeah, so most abortion medication would be based off progesterone antagonists, which is a key thing. So after, I guess, you have this idea of pregnancy, you'll have childbirth and the hormones associated with that. So progesterone maintains endometrium and inhibits another function of progesterone is actually inhibits labor and lactation. This means that if you have progesterone, you can't have milk. You can't let it inhibits prolactin from milk let down. Prolactin increases. Prolactin. Oxytocin will increase sometime in the third. And then relaxin and whatnot. And estrogen is there. So these are the main hormones. The fetus will have a lot of strains to the maternal anatomy and physiology on the pregnant person's body. It restricts lung expansion, compresses digestive organs, and also increases glomerular filtration rate, leading to more urination or more frequent urination, and it also increases respiratory demands. So, you can have the phases of, I guess we should say, labor. First is the dilation of cervix and propulsion into vagina. During this time, myometrial layer switches to single unit organization and expresses oxytocin receptors. So this is because single unit gap junction coupled organization leads to synchronized waves of relaxation and contraction, which are stronger in producing pressure onto the cervix. So yeah, just as is explained, you get these sort of these single unit organization or gap junction organized smooth muscle cells, and that leads to more better waves. And oxytocin receptors are important for positive feedback of uterine contractions. One key thing for the onset of labor is prostaglandin production. Prostaglandin production is key for triggering the onset of labor, and only when it's produced will you'll technically get labor happening. And labor is enhanced by oxytocin for the positive feedback loop by, for instance, okay, oxytocin, if we first have, let's say first step, we have mechanical stimulation of cervical tactile neurons, and then this leads to oxytocin release, oxytocin release leads to enhanced contractions, as contractions lead to increased mechanical stimulation of cervical tactile neurons, increasing greater dilation. There's a lot of other methods to induce or accelerate labor. One is you can use rupturing membranes, balloon catheter, or prostaglandin gel. Another thing is intravenous oxytocin only after regular contractions. You can't really give it before contractions because that wouldn't necessarily make sense to give it that thing. And then after the labor, and then if everything goes right, labor is in three phases. Dilation, where the cervix dilates for the baby. Expulsion, where the child is expulted or using uterine contractions and pelvic and abdominal contractions, it's expelled out. And the placental stage, where the uterine contractions dislodge the placenta. Some things might not go well though. You can have hypoxia for the fetus, uh, aspiration of amniotic fluid, uh, placental retention, strokes, postpartum hemorrhages, etc. And medical interventions can include forceps using labor assisted or C-sections. And the lack of progesterone we mentioned afterwards, it kind of spikes up and then down. The lack of progesterone after birth leads to essentially, um, and leads to, because progesterone inhibits the rise of prolactin for stimulating milk production. So I should say prolactin to milk production, then progesterone inhibits this. If we don't have progesterone anymore, we'll have milk production, and then milk production can begin. Estrogen helps here. Well, first, estrogen helped for, well, oxytocin, right? Estrogen helps stimulate oxytocin receptors in both the cervix, well, I guess you can say the uterus, and also in mammary glands. So what that means is once you have this sort of estrogen that's available, your mammary glands are able to secrete milk after just producing as well. So yeah, milk production will be done using the prolactin levels. You should also mention that milk production involves merocrine and apocrine methods of secretion. So merocrine, uh, I should say, involves the product being released through exocytosis. And then merocrine is shedding of apical portions. Apical shedding with vesicles. And then mammary secretions first begin with colostrum, which is protein-rich and antibiotics. And then it switches to breast milk, which has more fat, fat and lactose. It's more nutrient rich. You should also know it's low in vitamin D and iron, human breast milk. So formula is sometimes useful for that. And contraction of mammary tissue is in response to oxytocin leads to ejection. Let's see what else. Stimulation also leads to further and triggers reflexes that further lead to milk ejection and milk production. And yeah, this is basically all that's important. And I guess the next time you'll be hearing from me will be going over cardio. Thanks for listening.